Hello everyone. Welcome back to the new session of Biology, Standard 11th, Chapter 10, Animal Tissue. In the last video lecture, we studied the details of bone. In that, we studied about spongy bone and compact bone. Then we studied the details of muscular tissue. In that, we studied three types of muscles, that is, skeletal muscles, smooth muscles, and cardiac muscles. Today, we'll start with the last part, that is, nervous tissue. Let's see the introduction of this topic. Nervous tissue is composed of nerve cells, or you can also call it as neuron, and neuroglial cells. That means two cells are present in nervous tissue. One is neuron, other one is neuroglial cell. Now, neurons are the structural and functional unit of nervous system, whereas neuroglial cells are non-nervous and they are supporting cells. Now, these neuroglial cells actually they are filled in the interneuronal space. That means the space between two neurons. And these neuroglial cells are capable of regeneration and division. But if we talk about neurons, neurons are not able or they are not capable of a regeneration. That means they do not divide and redivide because they lack centriole. Now, intercellular matrix is absent in neural tissue. Now, coming towards functions of nervous tissue, first thing is that neurons they are an impulse generating and impulse conducting unit, and they bring about quick communication within the body. That means their action is very quick, or you can say very fast. Then you'll find neurons change action potential of their membrane on receiving any of the external stimulus. And this property of neuron is called excitability. And lastly, they also carry a wave of electrical impulse from dendron to axon. This is actually called as conductivity. So they are giving quick responses, they generate a conducting impulse, and then they are giving us the property of excitability as well as conductivity. Now, coming towards the next part, that is structure of neuron. If you see a neuron, it is made up of cyton, that is cell body. This cell body or cyton contains granular cytoplasm, which is called as neuroplasm, and it contains a nucleus which is present in the center. Then in this neuroplasm, you will find mitochondria, Golgi apparatus, a rough endoplasmic reticulum, and granules are present. The granules which are present in neuroplasm are called Nissel's granules, and they are made up of RNA. Then you will find cell body gives out two types of processes via dendron and axon. That means these two processes are called as dendron and axon. Talking about dendrons, dendrons are short branched processes. You will find fine branches of dendrons are present around the cell body, and these branches are called dendrites. If it is single, we call it as dendron, but you will find many branches are present, then we can call it as dendrites. And their main function is to carry the impulse towards cyton. That means whatever impulse comes to your body, that actually first comes to dendrons, and then dendrons passes that impulse to cell body, and then from cell body it enters inside the axon. Now, what is axon? Axon is a single, elongated, cylindrical process. It is bounded by exolemma. That means you can say the protoplasm of axon is called exoplasm, and the outer layer of exoplasm is called exolemma. It contains large number of mitochondria and neurofibrils. Axon is enclosed in a fatty sheath. That fatty sheath is called myelin sheath. Outer covering of myelin sheath is actually neurilemma. Myelin sheath and neurilemma, they are the parts of another cell that is called Schwann's cell. Schwann cell shows a nucleus at periphery. The myelin sheath is absent at intervals along the axon, and the place where myelin sheath is absent, that place is called node of Ranvier. And the last part, or you can say the terminal part of an axon, is called telodendron. Now, next part is types of neurons. Based on their functions, neurons are classified into three types. They are afferent neuron, interneuron, or you can say association neuron, and afferent neuron. Let's see the details of each. Let's start with first one, that is afferent neuron. These neurons, they carry impulses from sense organ to central nervous system, and as they carry the impulses from sense organs, so it is also called as sensory neuron, and it is found in dorsal root of spinal cord. Second one is afferent neuron. These afferent neurons, they carry impulses from central nervous system to effector organ, as they help in giving response with the help of effector organ. So they are also called as motor neuron, and it is found in ventral root of spinal cord. And third one, that is interneuron, or you can also call it as association neuron. They are located between sensory and motor neurons. They perform a processing and integration of sensory impulses and activate appropriate motor neuron to generate motor impulse. That means they help in interpretation of whatever the sensory neuron brings in, and then it changes that sensory impulse into motor impulse so that you can give proper reaction of an action. Now, depending upon the presence or absence of myelin sheath, neurons are classified into two types. They are myelinated, or you can also call them as medullated nerve fibers, and non-myelinated, or you can also call them as non-medullated nerve fiber. The one which is having myelin sheath is called myelinated, and one without myelin sheath are called as non-myelinated. And now let's see the details of these two, but we'll study it in the form of differentiation in order to understand it properly. Uh, you will find in myelinated, there is myelin sheath present, and in non-myelinated, myelin sheath is absent. Actually, this myelin sheath is nothing but they are insulating fatty layer. So this fatty layer is present in myelinated nerve fibers, and same fatty acid which is absent in myelinated, non-myelinated. So you will find sheath is absent here. In both the nerve fibers, you will find Schwann cells are present. But in myelinated, Schwann cells secrete myelin sheath, and in non-myelinated, Schwann cells do not secrete myelin sheath. Because myelinated nerve fiber has got fatty layer, so it appears white in color, and non-myelinated, they appear gray in color. You will find myelin sheath is not continuous and it is interrupted at certain positions. That parts are considered as nodes of Ranvier. So you will find node of Ranvier is present in myelinated nerve fiber, whereas nodes of Ranvier is absent in non-myelinated nerve fiber. Then a neurilemma. Neurilemma surrounds the axon in myelinated, whereas neurilemma does not surround the axon in non-myelinated. Then you will find conduction of impulse is very quick or fast in myelinated, whereas in non-myelinated the conduction is slow. Reason is that in myelinated nerve fibers you will find um, the impulse jumps from one node of Ranvier to another node of Ranvier. So that kind of jumping movement is called as saltatory conduction. As it shows saltatory conduction, so the conduction is very fast. Whereas in non-myelinated, node of Ranvier is not there, so it does not show saltatory conduction. Therefore, the conduction is slow. Now, best example for myelinated nerve fibers are cranial nerves of vertebrates. And for non-myelinated, you will find autonomous nervous system is best example. Now, there is a functional contact between axonal ends and dendrites of adjacent neurons. These contact between axonal end and the dendrites of new or next neuron is called synapse. Actually, it is a gap between the two neurons. This you will study in detail in standard 12th in chapter named control and coordination. So here you have to just remember the name, that is the gap between two neurons is considered as synapse. Now, based on the number of processes given out from cyton, neurons are classified into three different types. They are unipolar, or you can also call them as monopolar neuron, then bipolar neuron, and multipolar neuron. Let's see the details of each. Let's see the first one, that is unipolar, or you can also call them as monopolar neuron. In this, you will find there is a single process which originates from cyton. You will see that both axon and dendron, they arise from cyton at one point. Okay? And they conduct impulse to central nervous system. An example is you can see the neurons of dorsal root ganglion of spinal nerve. It is a unipolar neuron. Then bipolar neuron. It has two processes. That means a single dendron and a single axon. They are given off from opposite poles of the cyton, and they bring about transmission of special senses, like for example sight, that is whatever you see, smell, taste, hear, whatever these are special senses, they are done because of bipolar neuron. So example of this neuron is neurons of retina of eye or olfactory epithelium, etc. Then multipolar neuron. Here you will find cyton is star shaped and it gives out more than two processes. There is only one axon and remaining are dendrons. So we can say that dendrites are more here. Axon initiates from a funnel shaped area called axon hillock. And best example is neurons of central nervous system, and you can find it in Purkinje cells in cerebellum, etc. So let's see quick summary what we studied today. Today we studied the details of nervous system, that is nervous tissue. In that we studied about functions of nervous tissue, then structure of neuron and types of neurons. So we have completed the entire chapter, that is animal tissue. We'll continue again in the next video lecture with new chapter. Till then, take good care of yourselves. Thank you.